Most of us know someone or have a family member that has been affected by breast cancer. In fact, 1 in 8 women in the U.S. will develop breast cancer over the course of their lives. 41,000 women in the U.S. are expected to die in 2019 as a result of breast cancer, but this number has been on the decline since 1989 because of increased research and development into cancer treatments. Many treatments for breast cancer have come out of this, and one of the newest ones is Ribociclib, known commercially as Kisqali. But before we talk about how ribociclib works, we have to take a look at what enzyme it targets and how that enzyme contributes to breast cancer. E2F transcription factors are important for the G1S checkpoint in cell growth and division. These transcription factors allow the cell to move from the G1 phase to the S phase and begin DNA synthesis. In normal cells, the protein retinoblastoma 1, or RB1, is present, and it binds to the E2F transcription factors, which slows down the rate of reproduction of cells. When RB1 is phosphorylated, it releases the transcription factor E2F. RB1 is phosphorylated by cyclin-dependent kinases 4 and 6, also known as CDK4 and 6. In cancer patients, the RB-E2F pathway is dysfunctional, which allows the cell to continue into the S phase unregulated. For phosphorylation by CDKs to occur, phosphate groups from ATP molecules must be transferred to the CDK enzyme. This is followed by activation via cyclin D1 binding. Ribociclib acts as an inhibitor by blocking ATP clefts on CDKs 4 and 6. By the way, this is a CDK with ribociclib attached to it. Without phosphate groups bound to the enzyme, it can no longer phosphorylate RB1. This then prevents the release of E2F transcription factors to reduce the rate of unchecked tumor growth. Ribociclib includes many nitrogen-containing functional groups, such as amines, imines, and amides. There are also 3 aromatic ring structures, including pyridyl rings, a cyclopentane, and a cyclic diamine, that make this molecule hydrophobic. Several interactions are responsible for ribociclib's binding properties. The lysine and aspartate residues are important to the ATP cleft because they can hydrogen bond with the drug. Ribociclib, with the trade name Kisqali, is a new drug, only being approved in March of 2017. Therefore, only 2018 data is available. In that first year, Kisqali made $235 million. However, Kisqali has 2 big competitors that are also CDK inhibitors, palbociclib and abemaciclib. Although these drugs made $4.1 billion and $255 million in total sales last year, positive testing results are boosting Kisqali as it tries to catch up to the 2 other drugs. By 2024, Kisqali is predicted to be making $2.4 billion annually, when it will hopefully also be saving a lot more lives. Thanks for watching. Tune in next time for more fun with drugs.